Disclaimer. Recorded on June 20th, 2026. Information reflects the status discussed as of the presentation date and may have changed since recording. This recording is from ELJ-101 AAV gene therapy for the treatment of CDKL5 Deficiency Disorder, presented at the 2026 CDKL5 Family Educational Conference. It is provided for general educational and informational purposes only. As of the presentation date, the gene therapy referred to as ELJ-101 was investigational and in pre-IND development. The sponsor described receiving written FDA feedback on selected pre-IND development questions and characterized that feedback generally as positive. However, the specific content of FDA's feedback was not shared in this presentation. No FDA clearance, approval, licensure, endorsement, agreement, authorization to begin a clinical trial, or determination of safety or effectiveness should be inferred. ELJ-101 has not been approved, licensed, or authorized by FDA or any other regulatory authority for clinical use. This presentation is not medical advice, clinical trial recruitment, an offer of treatment, or a guarantee of timing, eligibility, access, safety, effectiveness, or availability. Development plans, manufacturing, possible sites, eligibility, dosing, timing, access, and future milestones are subject to change. Viewers should consult their treating physician, geneticist, or other qualified healthcare professional before making medical or clinical trial-related decisions. The presenter is affiliated with the investigational therapy developer and sponsor, Elaaj Bio, a wholly owned subsidiary of the Loulou Foundation. Official program details, when available, will be provided through official Elaaj Bio communications. The organizations are not responsible for incomplete, edited, unofficial, third-party, captioned, translated, summarized, reposted, clipped, or redistributed versions.
On behalf of Elaaj Bio, I'd like to thank IFCR for inviting us to talk today. And I'd like to thank all of you after a wild night of karaoke for getting up early on a Saturday morning and coming to this session. As I think probably everybody here knows, Elaaj is a wholly owned subsidiary of the nonprofit Loulou Foundation and was founded in 2018 with a sole mission, which was really to give CDD families and patients multiple transformative therapeutic options that would directly address the deficiency of CDKL5 in these individuals. So our lead program right now, ELJ-101, has focused on using a adeno-associated viral vector and parts that is basically composed of parts of a virus to deliver the CDKL5 gene directly to the brain. And this program was initially started by Jim Wilson and Elaaj Bio, and then was acquired by Elaaj for further development. So there were some excellent sessions yesterday that went through development of different therapeutics, and I think that a lot of people in this room are familiar with the fact that it is a long process and has multiple different parts to it. So where Elaaj is right now is we started off by building AAV vectors that we could use for research use, and built several that could be tested then in animal models of the disease. And then when we found that there was actually efficacious benefits from those different vectors, we chose one to make as our clinical candidate, and then performed both dose finding and safety studies in non-human primates to be sure that whatever we were going to bring into the clinic would have a chance of efficacy, and then would also be safe, at least from animal testing. So where we are right now is at the stage, as of June, is at the stage where we have begun manufacturing our clinical grade product. The good news is that we've received positive feedback from FDA regarding both our manufacturing process and our plans. This clinical or good manufacturing practice batch is officially underway as of this month, which is a huge milestone that we're very proud of. And production and testing should be finished by December of this year. Now, on the clinical front, we've also gotten some great news from FDA, and that they've given us positive feedback regarding our dose selection strategy, and also on the overall design of our first-in-human program. And we are currently at the stage where we are considering different sites, and we're evaluating sites in both the United States and in Europe for this first-in-human study. As many of you know, typically the first studies with a new therapeutic are focused primarily on safety and on tolerability. But because this is a gene therapy, we've also built in a number of different efficacy measures as well, because every single patient who is treated can generate data over the course of a study that can be used to determine whether or not this therapy is actually helping. So we're anticipating enrolling probably around 12 patients in this first study. The way that the dose is administered is it's a one-time dose into a fluid-filled area that surrounds the brain called the cisterna magna, which is found in the back of the head. And there'll be, the way that the drug will be administered is through a needle that will go in right at the top of the neck into that fluid-filled space, and then our viral vector will be infused into the cerebrospinal fluid. Now, I should say that as of about probably 50 or 60 years ago, this was a procedure that was performed at the bedside by neurologists. This was really before the advent of imaging studies. So this is a tried and true technique that we think will give good viral delivery to the entire neuraxis, the brain and spinal cord. The duration of the study will be approximately 1 year, and then all participants who complete this study will have the option of rolling into a 4-year-long long-term extension study where we'll be able to continue monitoring safety and also efficacy variables. The beginning of the study, right after being dosed, there's a lot of intensive visits because we want to make sure that we are really conducting good safety monitoring in terms of following labs and in terms of following a patient's clinical course. So for the first 3 to 4 weeks, there are several visits. After that, the visits will space out and be roughly every 3 months. And those visits that are later on in the study will be primarily focused on efficacy. In addition to safety and tolerability, the specific things that we'll be looking at from the efficacy standpoint are seizure frequency and severity, cognition, communication, motor skills, adaptive function and behavior, quality of life, and sleep. And I have to thank the community for being very collaborative. We've talked to both a number of clinicians who are very familiar with CDD, and then also a number of parents and patient advocates who have advised us on the types of things that will make, hopefully the most difference for your children, and that also will be measurable in the course of a clinical trial. And we anticipate that if everything goes smoothly with our manufacturing and smoothly with the FDA submission, that we should be able to begin enrollment sometime in the spring or early summer of next year. As I mentioned at the beginning of the talk, Elaaj is committed to developing not just one, but several different transformative therapies for CDD. So there are a number of other avenues that we are also currently exploring that include things like targeted X reactivation. As I think you probably all know, girls have 2 copies of the X chromosome. One of them gets inactivated over the course of development, and in patients with CDD, that is often the copy of the X chromosome that has a normal copy of the gene. And so we are collaborating with investigators to try to figure out how to maybe reactivate that normal copy of the gene and accomplish gene therapy using a patient's own DNA. We're also pursuing approaches that involve novel AAV capsids for different ways of delivering of the CDKL5 gene editing, antisense oligonucleotides, and non-viral delivery of different therapeutics. And the hope is that we will have then a whole armamentarium that can be brought to bear on the disease and that will take into account variability between patients so that there are solutions to multiple different aspects of the disorder. I kept this talk short on purpose because I wanted to make sure that you all had time to ask questions. But I, again, really want to thank the community, families, caregivers, patients, advocates for their collaborative view of helping us get to where we are right now, and for everything you've done in terms of being enrolled in studies like the CANDID study and the ICCRN study. That natural history data is going to be invaluable when we have to, number one, educate regulators about how this disease actually progresses, and number two, when we do start to generate efficacy data from our clinical trial, that we'll have something to compare to. We've also been very lucky in that we've had a number of different industry and academic partners and collaborators, from many of the clinicians who are here to other sponsors who have been very generous with sharing their protocols, their approaches, and their own experiences as we've tried to develop our program. Thanks all for your attention. I'm happy to take any questions that you have. And if you don't have a chance to ask the questions now, please come and find myself or Russ. Over the course of the day, we'll be happy to chat with you. Thanks.
Have you decided how you're going to? It's me.
Oh, no, thank you.
Here I am.
It's tough up here. There's so many people.
Is there any kind of, so have you decided how those initial 12 individuals will be chosen?
Yeah.
And is there any restrictions on male versus female?
Yeah, no, great question. So what we're thinking is that for this first study, it will likely be girls who are ages 4 and up. There's a couple different reasons for that. One, the overall disease is potentially a little more homogeneous in girls versus when you bring boys into the equation, and there may be, from a safety standpoint, reasons to also start in girls. 4 and up, because brain size by the time you're 4 is pretty close to adult brain size, so the dosing of our viral vector can be the same in all of the individuals that are in this study. Now, I want to mention, though, this is a pilot study so that we can make sure that this is a safe approach and that there is some reason to believe that there will be benefit. Elaaj is committed in further stages of the trial, if things look good here, to including and being as inclusive as possible. So including patients who are down to maybe 6 months of age, and including boys in separate arms of whatever subsequent trials we do.
And will all of those, the 12, be within, you said you're looking at sites for United States and Europe. So is it determined like how many within the United States, how many within Europe? And sorry, I got one more I'm just gonna ask because I know everybody's gonna have questions. What about the medications that our children are on right now? Like once, you wanna measure, seizures and sleep and all of that, but all of our kids are on medications to help control those right now. So, and you can't take them off right away. So how is that going to be determined with weaning off of medications?
I sort of feel like I need some help with the clinical development. Are you available for being hired? Because these are the kinds of questions that we ask. So we would like to enroll patients at all of our sites. That's one of the things that we would really like to do, and we'll just have to see how fast we can get sites activated, how regulators in the different countries are going to be responsive to what it is that we're trying to do, and so we'll sort of cross that bridge when we get to it. But best case scenario, we would have at all of the sites that we open, we would have individuals. In terms of some of the inclusion criteria and medications, as long as the medications that are being used are stable for some period of time, depending on what they are before the course of the study, someone can be enrolled. And if there's a need, like let's say with seizure medications, that now the medications that a particular child is on are no longer effective and something needs to change during the course of the study, we understand that. We just have to note that that's happening so that we can take that into account when we're doing the data analysis later. So we're trying to be as inclusive as possible in the folks that will be eligible for the study.
Is there a plan, though, to wean off the medicines once the trial has started, or would you rather keep everything the same?
One of the things we'll be looking at is seizure frequency and severity. If we see that seizure frequency is going way down after treatment, and there is an opportunity to wean medications, that's actually one of the endpoints that we'll be looking at, is what is the overall anti-seizure medication burden for our patients, because that's one measure of whether or not we have efficacy with this therapy.
Thank you again for all the work you guys are doing. It's incredible. Can you just expand on the positive feedback you're getting from the FDA, specifically what you're seeing in the trials and treatments with the, animal testing?
So FDA has not reviewed all of our data yet. We've asked them some very specific questions about our manufacturing strategy and about our clinical strategy, and they have been supportive of our overall approach. So the next step for us is to submit what's called an investigational new drug application, which we anticipate will go in sometime probably in late fall of this year. And at that point, they'll have the opportunity to look at everything that we've done, all of the data that we have generated on the manufacturing front, all of our non-clinical data, and in detail what our clinical plans are, and then they'll be able to comment on what it is that they think is okay and what they would like us to change or include or what they might want us to exclude. So at this point, we have very positive early feedback from them. The rubber is really going to hit the road when we submit that IND, and then we'll have much more detailed discussions with them.
I have a question about the nature of gene therapy, because it's a one and done right now, right? So for these 12 initial kids, what are your thoughts? Are you also doing any dosing measuring, or is it just safety and tolerability? And if yes, what happens after for them, since they lost the chance to another treatment?
Yeah. So all of the doses that we're considering are doses that, based on the animal studies, we believe will be efficacious. We're not proposing to use any doses that we think would just show safety and tolerability without a chance at efficacy. And that within the gene therapy community, there have been a lot of discussions about the ethics of doing that, because as you point out, this is a one and done therapy, and as of right now, we can't redose. So it's very important that every single participant in a trial like this have the opportunity to experience benefit. And the doses that we are considering are within the range that we believe will give them that chance.
So other than safety and affordability, are you going to learn anything else dose-wise from that that
So the doses that we're considering, depending on how our discussions with FDA go, we're considering either 1 or 2 doses. And again, all of them should be within the range of efficacy. Based on what other gene therapies have done, we think that we're in the range where things should be good. But to your point, if we do end up doing 2 doses, we can then do a comparison of those 2 to see if one might be more efficacious than the other. Yeah, that I've spent the last 10 years doing rare disease research in industry for different sponsors, and it's a really hard question to answer in rare diseases, because typically the number of patients you can even identify and you can enroll, and the amount of time that you can realistically watch them in a clinical trial is so short that often you can't use the kinds of statistics that one would use if you were doing like a blood pressure medicine trial or a diabetes trial. So there are much more qualitative measures that you end up using to try to get an idea of whether or not there is actually a dose response.
Anyone
Hi.
else have a question?
Are you open to discuss other sites' participation as Latin America, Canada, MENA, or is it a closed decision?
Yeah, so for this first study, I think that we've identified sites that are centers of excellence, very experienced PIs, who not only have experience with CDD, but also have experience with gene therapy in general. Our goal, though, is to, after this pilot study, really, is to then expand, and we will be considering areas all over the world at that point, assuming that we can move to that next stage.
Yeah. So you said eventually possibly go to children as young as 6 months. So in the timeline of that, so you do the initial and then you test the efficacy and all that stuff.
Yeah.
So like how long would that be before you would know if that's even a possibility that you could do that?
Like, clinical trials don't move fast enough for any of us, honestly. If I had to conservatively say that maybe it would take a year to enroll this first study, and then we have to have a year of data before we can really look at it and have enough patients dosed that we think we understand the safety and the efficacy portions. And then at that point, go back to regulatory agencies and negotiate with them. We're probably talking somewhere 2.5 to 3 years, best case scenario. What I can tell you, so I've been with Elaaj for about the last 9 months. Between Elaaj and Loulou, the people there are very energetic, very intelligent, very driven, and very focused on this mission. And we are trying to do things as efficiently and as quickly as possible so that we can bring a safe and efficacious therapy to as many people within the population and the community as possible.
So what would be your ideal first patient? Be uncontrolled seizures or controlled seizures with meds?
My ideal first patient would be one who would completely respond to the therapy.
Well, true. But
I think that because seizures is going to be one of the endpoints, that it is highly likely that there will be a cutoff number of seizures that that individual has to have over the course of a 28-day period. Like if any of you participated in some of the other anti-seizure medication trials, there are typically these cutoffs that you have to be above so that you can actually see whether or not there would be a lowering in that frequency. We're still kind of deciding what that cutoff would be and what types of seizures based on what regulators will accept as a potential endpoint to decide that. But we realize that CDD is a highly heterogeneous disease from the standpoint that there are people who have hundreds of seizures a day and there are some people who have no seizures, and they may be affected differently in terms of their cognition or their communication or their motor skills. And that's why we have a very broad set of endpoints that we'll be looking at as part of this trial.
Hi. Just to follow up on the gentleman's question, I'm wondering if you guys spoke to the regulatory agencies about the potential of a midway interim analysis, and then you could determine efficacy preliminarily based on that.
Yeah. So this first trial, I should have mentioned this, the first trial is going to be open label. There's not going to be a placebo arm or a sham arm at all. So people like myself might be looking at the data every day to see what is coming in, and we will be able to follow that very, very closely. And in our statistical analysis plan, we've already talked about looking at the 6-month time point and the 9-month time point and the 12-month time point, to be able to see whether or not there are any indications of efficacy. There are some confounding variables, as you probably know. With gene therapies, we typically have a period of immunosuppression that goes along with the gene therapy because it is a virus that you're administering, and the type of immunosuppression that we're considering is corticosteroids, which we know actually has some effect on some of the seizure types that kids with CDD experience. So we will have to get far enough away from that immunosuppressive period to make sure that there's no confounding from that if we're looking at seizures as an endpoint, for example.
No.
Any other questions?
Any specific medications or devices that would exclude you?
In terms of other types of therapies, any prior gene therapy would be an exclusion. And being within a certain period of time of other investigational drugs of some sort will also be an exclusion. If you've taken part in other clinical trials with other types of investigational drugs such as ASOs or other small molecules, there will be a time period during which you would not be able to enter this study, just to let those things clear out of the system so that there's no confounding from their effects.
Can I ask a quick question about the burden of visits? Because you said there will be a lot of visits at the beginning, and
Yeah.
I wonder, will all of them be on site, or will
Yeah.
any of them be at home? No, all on site.
So our thought is for the first 3 to 4 weeks that we would like participants to be very close to the hospital for 2 reasons. One, there's going to be a lot of blood draws because we want to be following, there are known safety implications of getting gene therapies, and we can follow those through different types of laboratory testing. We want to follow those very closely so that if anything does start to become untoward, we can deal with it right away. So we're thinking about ways of having people stay close to the
Kinda
hospital.
move
That's right.
to the site and stay there
That's
for a
right.
month or something like
That's
that.
right.
Got
That's
it.
right. And then after that, once we're sort of out of the woods that first month or so, then people will be able to go back home and then commute in for their roughly every 3-month visits.
Got it. That helps a lot. Thanks.
Those first 12 have the same therapy, or will this therapy be tailored to their specific mutation?
Right now, there are no exclusions based on mutation.
And will those 12 have the therapy at the same time, or it might have, we'll see how 1 or 2 react and then follow up with the others. So
the thought is for the first 3 individuals who are dosed, we'll dose 1 patient and then wait 2 months, and then dose a second patient and then wait a couple weeks, dose a third patient, then wait another month, and then have what's called a data safety monitoring board meeting where independent experts in this field will look at all of that data along with us to make sure that things are safe and that there is reason to continue with the study. And in that way, if there was something that in some way was dangerous, we would identify that very early before exposing a lot of other individuals to the treatment.
So I just want to ask, if everything goes well and it becomes in clinical use, how will it be funded? How will the gene therapy be funded for everyday use?
Oh, that, boy, I don't know if I'm qualified to answer that. I think that so if, Helen, if we get to the point where this is great and it gets approved and then we're talking about commercialization, I think that the answer really depends on where you are, right? I mean, things are very different in Australia than they are in the United States, than they are in Europe, than they are in South America. So there are a number of different considerations there. In many places, that reimbursement would be negotiated with the government because they are, with socialized medicine, they are the single payer. Places like the US, there would be negotiations with individual insurance companies and also with the government in terms of Medicaid, for instance. But it just really depends on where it is. And in terms of things like pricing and all, we haven't even thought. The only thing we're focused on right now is really, developing this treatment. And I will say that as someone who's been in industry for a long time, it is very refreshing to work for a company that is a wholly owned subsidiary of a nonprofit, because those types of questions are maybe not quite as important, and we can focus more on the important stuff.
I have a question. Just going back to me again.
That's right. You're going to send me your resume, right?
Absolutely. Just going back to the male versus female, just trying to understand, can you elaborate a little bit on specifically why you feel like there would be a little bit more of a fear in dosing males versus females? I mean, I understand the X versus the Y chromosome and all of that, but we're not messing with the current X or Y chromosome. We're giving a healthy one to all individuals, male or female. So what is the specific scare there, I guess?
I think it's really a couple of things. One is we don't have as much natural history data to completely understand how the disease sort of progresses in males, and so we don't understand as much about some of the other comorbid factors that males might have, and it's just because there are so many more girls who are affected than boys. Also, at least historically, we've thought that the disease is more severe in males, and so the degree to which we might have to restore CDKL5 function is potentially higher, and we don't know how dialing that up that much might actually affect them as opposed to girls. So for those reasons, and also because we have that more data really and on the homogeneity of the disease in girls, that's why we're deciding to start there. But as I said, I think this is not atypical for a first-in-human study where the population that one enrolls in that study is much more focused. And then it's once you've got proof of concept that something is safe and that it works, then you expand that population to try to more broadly, reach the community.
If your child has a mosaic mutation, does that disqualify them or affect this at all?
I don't think we have that as an exclusion, but come find me after this is all done, and I'll take a look at the protocol.
A follow-up question is there a maximum age? I know you're looking at minimum 4 years, but
right now there's no maximum age. It's just 4 years and up.
Any more?
I know we talked a lot about, others have asked questions about medications and so forth. So my granddaughter is due to have scoliosis surgery in the fall. So when you're talking about testing and MRIs or those kind of things, does that interfere with any of that?
There is it should not, and there is a time period after major surgery, during which we wouldn't want to enroll someone just because they just underwent a huge stress with that surgery, and then to be putting them through an additional stress from a health standpoint, probably not the best thing. But after a certain period of time, going out from a major surgery, people will be eligible.
Quick question about a follow-up first to the VNS DBS thing. So would you say that then kids who don't have that and are waiting for gene therapy maybe should hold off on that in anticipation of needing this kind of imaging?
So what I would say is when there are tried and tested therapies that are known to work in patients, in my own opinion, you should always do those because there's evidence that they work. One thing that is part of drug development, we always think it's the shiny new thing, and we always think, we think about the positives. But those kinds of trade-offs against different therapies that we know work, I think that that's a very individual discussion that you have with your physician and you think about, because if there's a great need for something that works right now, even if it would exclude you from a therapy that you're not sure is going to work, it's often better to go ahead and do that, in my opinion.
And is this a permanent exclusion or just for the trial, do you think? And actually the same question goes for all the other things you mentioned, other trials, which you just mentioned gene therapy, but I'm wondering about investigational drugs for epilepsy, for example, if somebody was in the Fintepla trial or ganaxolone trial, is that an exclusion just for the...
So for investigational drugs that will clear and get out of your system.
Okay, so it's just the, right, I know.
Yeah.
Got it, it's only if it's still investigation.
We've been recently on a new clinical trial for someone who's not yet FDA approved.
Got it,
And they're still in an experiment.
Yeah.
But if you were in the past, it doesn't exclude you. Got it.
And even if you were in a trial for something that hadn't been approved right now, after a certain period of time, what we would say 5 half-lives of that drug, then it's out of your system, then you'd still be eligible. In terms of the exclusion criteria for trials are very focused, because again, we're trying in some way to have a more homogeneous population or look at a population that we think has less of a chance of having harm based on the trial. When we start to generate data, that's when we can become more inclusive, because we understand more of how that drug is going to work, how the approach is going to work, and how safe it's going to be for people. So ideally, we will develop a therapy that will be of broad applicability for as many people who have CDD as possible. That's what we're aiming for. But often in drug development, you have to start more focused and then expand from there.
Thank you.
Is there going to be some kind of cap on severity of the patient? Because a lot of our kids, there's a very wide spectrum. Most of them have seizures, but there's a wide spectrum on how those seizures affect the child. So are you going to look at a patient and be like, they're too severe or they're not severe enough, and take that into account?
So as of right now, for seizures, for instance, there's no cap on number of seizures or severity of seizures. For overall severity of the condition right now, there's also no cap. And it's something that we are discussing with with clinicians and with some of the PIs to see whether in this first study it might make sense to have some sort of ceiling on that, but as of right now, there's not one. And the reason for that is that CDD is a neurodevelopmental disorder and not a neurodegenerative disorder. So in the past, I've worked on different diseases that are equivalent to childhood Alzheimer's disease, for instance, where you are losing parts of your brain over time. CDD is not like that. And so there's reason for us to believe, based on animal studies and based on the way we think we understand the disease working, that there may be opportunity for benefit regardless of how old one is. And it might be that benefit is greater when you're younger than when you're older, but thinking about the type of dysfunction that lack of CDKL5 causes, there is reason to believe that even people who are older might derive some benefit from an approach like this.
Okay, I'm going to ask a question. Do you have plans to test for exposure to the virus or seropositivity to seeing if someone would have an immune response to the viral vector.
Yep. So we are going to test for that, and we're going to monitor it. It is not an exclusion criterion. And there have been a number of more recent gene therapy studies that have shown that it really doesn't matter if you've been exposed to the virus previously, particularly when you are administering the treatment directly into the central nervous system.
Okay. So I think we will, thank you, Steve, for Dr. Merzich for doing that.
Disclaimer. Recorded on June 20th, 2026. Information reflects the status discussed as of the presentation date and may have changed since recording. This recording is from ELJ-101 AAV gene therapy for the treatment of CDKL5 Deficiency Disorder, presented at the 2026 CDKL5 Family Educational Conference. It is provided for general educational and informational purposes only. As of the presentation date, the gene therapy referred to as ELJ-101 was investigational and in pre-IND development. The sponsor described receiving written FDA feedback on selected pre-IND development questions and characterized that feedback generally as positive. However, the specific content of FDA's feedback was not shared in this presentation. No FDA clearance, approval, licensure, endorsement, agreement, authorization to begin a clinical trial, or determination of safety or effectiveness should be inferred. ELJ-101 has not been approved, licensed, or authorized by FDA or any other regulatory authority for clinical use. This presentation is not medical advice, clinical trial recruitment, an offer of treatment, or a guarantee of timing, eligibility, access, safety, effectiveness, or availability. Development plans, manufacturing, possible sites, eligibility, dosing, timing, access, and future milestones are subject to change. Viewers should consult their treating physician, geneticist, or other qualified healthcare professional before making medical or clinical trial-related decisions. The presenter is affiliated with the investigational therapy developer and sponsor, Elaaj Bio, a wholly owned subsidiary of the Loulou Foundation. Official program details, when available, will be provided through official Elaaj Bio communications. The organizations are not responsible for incomplete, edited, unofficial, third-party, captioned, translated, summarized, reposted, clipped, or redistributed versions.